Hi everyone, very good evening guys. Question 1 over here was a very lengthy one, but first I'll give you the answer. They said which microscope enables quantitative measurement of the chemical constituents of the cell. See, a cell is made up of what all? It is made up of lipids, proteins, carbohydrates. So whenever you want to measure the chemical constituents like all of these that I've mentioned, you will be using something known as interference contrast microscope. Simple ways when you want to interfere, like your relatives interfering in your life, wanting to know every detail of your life, that when will you get the degree, what rank did you get, what marks did you get. Same way, you are interfering in the cells and you're wanting to find out how much of chemical, which chemical does it have. So the simple answer to this was interference. But if I go back to the clinical history, although it's not relevant, but what did you make your diagnosis over here? You had some bacterial infection. It was a respiratory disease. Then they said that there's a special culture which is having blood cystine and potassium tellurite. Potassium tellurite, and whenever tellurium comes, it gives black color colonies. This was actually a case of Corynebacterium diphtheria, because yes, that has the typical bull's neck and the pseudomembrane. Maybe you'll get questions tomorrow on this, and that is to do with the diphtheria, pseudomembrane, bull's neck, respiratory infection. This selective media for Corynebacterium diphtheria is potassium tellurite agar, and that always gives black color colony. So the history might have been very different, but the question was on a microscope. And if you see any, let's say if a regular light microscope would have been used, what is the appearance of Corynebacterium diphtheria? The Chinese letter appearance. So do you see Chinese letter appearance on interference contrast microscope? No, I see Chinese letter appearance on a regular light microscope. Although there was no question of light microscope in today's class, in today's test, but maybe you know the questions that will always come. Either they will give you a question which is to do with the labeling, so the TNT just does not stop at the question. It's about knowing the topic, and that's what we are going to do. Quickly letting you know what is the main hero of the story, the stage. This is the stage where you're going to keep this slide, and then where is the light present? This is where the light is present. Then light will go in all directions. I will want that no light should get condensed only over here. So there comes the most important question. Where is the condenser present? It is present below the stage. Meanwhile, everyone who said that other than Chinese letter, cuneiform appearance is also for diphtheria, and you're perfectly right on that. Coming back, condenser is present where? Below the stage. Now talking about the lenses. See, it's INI-CET, they will like to test you on lab oriented. Like in pathology, I was teaching you those flow cytometry, TEG, immunohistochemistry. Those are lab things. Same way over here, microscopes, stains, getting into the lab is what they'll ask you. Now coming to where all are the lenses. The lens where you are sitting near the eyes is the eyepiece lens. The lens which is closer to the object is known as the objective lens. And tell me their magnifications. For eyepiece lens, what is it? It is 10x magnification for this lens over here. And these 4 lenses are depending on, like you have zooming ability in your camera lens, same way over here as well. Scanner is going to be 4x, then low power is a little more, that is 10x, then next is high power, that is 40x, and oil immersion lens is 100x. So in a simple way to learn it, 4, 10, 40, 100, 4, 10, 40, 100. So now of course the question is, what is the total magnification? I will multiply these two, objective into eyepiece. So what is the maximum magnification that I can get? Eyepiece is always 10 and objective maximum is 100. So 100 into 10 makes it a 1000x magnification, which is maximum. These kind of questions are not coming, the one-liner ones. They'll give you a more applied question. They'll say there is a microbiologist. He is trying to find out some inclusion bodies or some bacteria or some parasite, and he is currently working on a 40x objective lens. When they say 40x objective lens and they ask me what is the total magnification, now what your answer becomes is whether you know that you have to multiply them. 400 times becomes your magnification if you're using this lens. So they'll tell you which lens is being used. Anyway, this was the basic. I think we have settled this interference contrast. Maybe there was another question on microscopes. As I said, don't go by this number. I've changed the order for discussion. What is the diagnostic method for this image? Obviously, if you're using dark field microscope, you will get a dark picture. But is that the question? This I picked up from your Q bank because I wanted you to be familiar with your PYQs first. PYQ to ho gaya. But now my next question is, they'll ask you in INI, how did you get a dark field microscope? And listen to this very carefully. To get that dark background, everything is similar to a light microscope. If you look very carefully, this is the stage and this is where you have kept your slide. Then ma'am, below the stage I have the condenser also. Then here I have the objective lens, maybe 10x, 40x or 100x. Everything is the same, but how have I made the background dark? Because now the light that I'm using is used as reflected light. I'm using the typical light bulb. यहाँ पे देखो same वाला light bulb is there, but I created a dark background by adding something called an annular diaphragm. It's like a block. I'm blocking some part of the light. That is why it is becoming, the final result is becoming black and dark over here. So they will ask you what is the reason for getting a dark background. Annular means like a ring, and diaphragm means a partition, right? Like we have the diaphragm in our body. Same way, diaphragm is a partition over here. Annular because it's like a ring. Now do you think it will be like an obstacle? Ab to light nahi jayega, light won't go forward. So now the light is only going from the peripheries, and that is why I am only getting some light and some areas are not, and hence the dark field. My question is, when will you use this? Whenever I want to see this special bacteria. What is this spiral bacteria called? Spirochete. Why can't you see spirochete on a light microscope? Because spirochete is very, very patla. See, I had to zoom in so much to show you. So it's very thin. Anything that is very thin, like a spirochete or maybe a flagella in a bacteria, I will be using dark field microscope. Over here spirochete photo was given. The answer was pretty obvious. Now you know why dark field, because of that annular ring, and I'm just going to change it. Now look at the next microscope. In the same, let's go order wise. It's the same light microscope. This is where your eyes are, ma'am. This is where the stage is, where you've kept your slide. Below the stage you said there will be a condenser. Then here is a light. Baki sab same hai. This is a light microscope, but this is not a light microscope. I can see the annular diaphragm. Which microscope does it become now? If I do +1, I added an annular diaphragm. Till now I think it is a dark field microscope now. But then I watched further, and near my eyes also you have added one more annular called annular phase plate. If only annular diaphragm would have been added, it's a dark field microscope. If annular diaphragm and annular phase plate has been added, now it becomes a phase contrast microscope. And that's the next question that you got in your test. In your test, you were asked that which microscopic technique will be used if you are wanting to generate a high contrast image. High contrast, ma'am, you can easily generate. For example, the background of this slide is what? The background of this PPT is white. So if I would have also worn white color clothes, then you would have not been able to see me, right? So I created a contrast that, okay, if the background is white, then I should wear something dark. That is how the contrast will be there, and that is how on the camera you will be able to see me. So usually we follow this, right? Why are we adding pink, blue, green, yellow colors in Path and Micro? To create a contrast. But here they said the contrast image is on a transparent viable bacteria. You have not added any color, no pink, blue, green, yellow of Path and Micro. And keeping the bacteria transparent, you are still able to see it. Then you can only do that if you change the contrast. Like while clicking a picture on your camera, you change the lighting and the contrast so that the image becomes better. So this means I'm playing with light. I'm playing with the phases of light, and that is what I will do using phase contrast. When I want to see transparent living bacteria, how did I play with the phase of light? Because I added two things over here. So if I just basically ask you, light jab ja raha tha, to light was all, can you see they are all, rays are in the same phase. But now what I have done, I have changed the phase. I have changed the rays of the light. By changing the phase of the light, I have created a contrast for use. So phase contrast microscope, where will I use this? Whenever I want to see internal details. Imagine this is a cell or bacteria and you want to see what is inside it. So ma'am, light change karo, light ka contrast change karo, you will be able to see that. But where in pathology do we use phase contrast microscope? Remember hairy cell leukemia, if you want to see hairy cells nicely, best way to see a hairy cell is going to be phase contrast. So I think this question is also settled for all of you, and I think most of you, if non-viable mention, they won't usually do that. Because if you're keeping, see the point is, if you're keeping the bacteria transparent, means you are not adding any chemicals, no ethanol, methanol, red, blue, pink color. So then on keeping it transparent and without chemicals, it shall remain viable. It shall remain viable. So there is no concept of a transparent non-viable bacteria that usually exists. Got that? So we are done with phase contrast. ये वाला भी देख लिया. What is this polarizing microscope? Where in pathology did I teach you? Apple green birefringence on a polarizing microscope was used for what? It is used for gout, is used for amyloid. But I was going to ask you gout, but now that I've said it on my own, where else can you use? Maybe in rheumatology that you study in medicine, and that is to do with polarizing microscope when you're differentiating gout versus pseudo gout. So now I want to ask you, I mean, this is very basic. You're going for INI-CET. No one will ask you, gout shows you uric acid increase and pseudo gout. So we'll write it. Gout to shows you uric acid crystals, which are going to be more, of course, and pseudo gout shows you CPPD crystals. But now I want to ask you that on polarizing microscopy, which is negatively birefringent and which is positively birefringent. So now very good. So pseudo gout में जिसमें PPPD crystals दिखता है, over here they are positively birefringent, whereas gout में uric acid वाला they are going to be negatively birefringent. Whenever you are checking for refringence, like in amyloid also apple green birefringence, for refringence we had the polarizing microscope. So this is also settled. एक और बचा, ये electron microscope के भी photos देख लेते हैं. The one microscope that looks like this spaceship, which is enclosed from everywhere, like completely enclosed. Why? Because over here, ma'am, you're not using light, you are using electrons ना. तो electrons जब use करोगे, you will have to maintain a vacuum. You can't keep electrons in the air, it's not going to work only. So for light microscope, you use light, and that is why you can keep the light microscope here on this table, because air will not interfere with it. But for electrons, you will need a vacuum, hence you closed it like a spaceship. Few questions from path and micro, very good, coming up over here. Quickly tell me, if you want to send something as a fixative, you want to send a biopsy for the path lab, if you're sending it for light microscope, which solution will you send it in? Formalin. These are like typical INI-CET fixative wala questions. For light microscope, you are using 10% neutral buffered formalin. For electron microscope, 2 to 2.5% of glutaraldehyde is going to be used. Very good. After you send it to me, now that you've sent it to my lab, now everything is my responsibility. Let's go back to path. Let's say you sent me a kidney biopsy, because kidney biopsy mein you have to do light microscope also and electron microscope also. So you sent me 2 kidney biopsies. One was in formalin, one was in glutaraldehyde. I will take that kidney biopsy and now place it where? If I have to do light microscopy, I will put that in a block of paraffin, wax. I will basically solidify that tissue inside wax, because wax works well with light microscope. But if I have to look at it under electron microscope, now I will put it inside resin. These are the embedding media. Where are you going to place the tissue? Okay, tissue has been placed, tissue is safe and sound. Now you have to look at it under microscope. On which kind of a slide will you see? Light microscope, so we know, ma'am, we used to use those glass slides in our practicals. So light microscope mein glass slide use hota hai, but glass with electron doesn't work well. So for electron microscope, I will prefer a metal slide, usually that is made up of copper. I think all of these are done, and now I can go back to the last microscope जब इतना कुछ पढ़ ही लिया था. So this question also I gave you, that which of the following best describes the process of fluorochroming? Fluorescent microscope, obviously. Chroming means adding some color. You must be adding all those fluorescent dyes, ma'am, pink, blue, green, and using fluorescent microscope. In fluorescent microscope, do you have unstained samples? No, I am adding some dyes. Are you applying some heavy metals? Not at all. In fact, do you know where do I apply heavy metals? In electron microscope, because right now you said that for electron microscope, ma'am, you are using electrons. Okay, let's assume this is a kidney biopsy. Electrons are coming and striking it. तो मुझे दिखेगा कैसे? Till the time I don't create a contrast, I will not be able to see it with my eyes. So sometimes in electron microscope, I add some heavy metals like uranium, etc., and they will create a contrast. So this is not for fluorescent microscope. This is also ruled out. Are you going to manipulate in fluorescent microscope with polarizers? No, polarizers to polarizing microscope ka hoga. Even by ruling out, I have option B as the answer, where dye will be used and there will be difference in wavelength. So everything about fluorescent microscope over here, if you look carefully, this also looks like a light microscope. This is where your sample is, ma'am, one objective lens, one eyepiece lens. Dekhne mein bhi light microscope, but the difference is the type of the light that I used over here, and the type of light is ultraviolet light. Then I saw a special mirror being kept over here, very famous dichroic mirror, and then I saw the light is going First it is going to the object, the slide, TB, malaria, jo bhi hai, and then the light is going back to my eyes. So the light that was coming initially was of a shorter wavelength, and then the light that I sent from my sample was a longer wavelength. And that's the question, that you have added some dyes in which they will re-emit the light at a different wavelength. Principle, simple words, shorter wavelength converted to longer wavelength. कौन सा वाला dyes? Very good, you will now start answering बस 1 2 3 4. पहले तो ये flow cytometry वाला dye I'm sure you guys remember, and that is FITC. Remember this, I taught you twice in both the days of the path TND. Fluoroisothiocyanate for flow cytometry. Remember, flow cytometry was for all those CD markers. Then we had two types of graphs. One was wo dot 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 wala graph. What is that known as? The dot plot. And then one flow cytometry graph was looking like this, which was known as histograms. Wo chhod do. Now let's come to the micro part. For TB, what are the fluorescent dyes? Auramine, rhodamine. I hope you remembered auramine and rhodamine because we are dealing with MTB. You did not think of rhodanine because that is going to be for copper. Now coming to malaria. Malaria का fluorescent dye acridine orange and fungus का calcoflower white. इसके तो नाम में ही fluorescent है. But now I have another question for INI exam. If in malaria, वैसे तो malaria we see under the usual stains, usual microscope. Let's assume you are seeing malaria under fluorescent microscope and you are using acridine orange dye. What is this technique known as? For malaria, if you are using the acridine orange dye, it is known as the Kawamoto technique. This is also a malaria question because usually we keep focusing on the usual malaria slides, banana shaped and all. If they use fluorescent microscope, acridine orange, now you call it as the Kawamoto technique. I think we are settled with this much. Now I have a question. I added all of these colors, that is why they were shining so green. Can you now tell me three things where you will not add any color mentioned over here? You will just keep that under ultraviolet light and it will start shining on its own. Things that show autofluorescence. Firstly, the two Spora brothers, Isospora, Cyclospora, coccidian parasites mein padhte ho. You will have a question today. So Spora brothers can shine on their own. You don't have to add any of thing. You only put them in ultraviolet light, they will start shining. Second, formalin also shines. But ma'am, I have a question. Here, if you ask me, on electron microscope, let's start with this. For light microscope, you were sending me the sample in formalin, right? Let's assume you are an intern in the dermatology department. I mean, no one goes to the derma department, rarely during internship, but let's assume you went for your internship to the dermatology department. They are doing skin biopsies. We know in dermatology you have bullous disorders. You have studied how you see the fishnet appearance and all those linear patterns and so many patterns that you see in bullous disorders. So for seeing those patterns, you have to send it for fluorescent microscope. Now if you send me the skin biopsy in formalin and I am saying that formalin has a property that it will shine on its own. So formalin will also shine and then that fishnet appearance will also shine. How will I get to know whether it is real type of fishnet appearance or whether the appearance is only because formalin is shining? So my question is, if you are sending me a skin biopsy for fluorescent microscope, should you be sending it to me in formalin? Answer is no, because formalin will start shining and then I will never get to see the real fishnet appearance. So our skin biopsies for immunofluorescence sent in formalin or any biopsy for fluorescence sent in formalin, is that the preferred thing? Answer is no, because formalin khud se shine karta hai. Spora brothers also shine on their own and NADPH also shines on its own. Let me give you an applied question for this. NADPH कौन से pathway में बनेगा? It is that G6PD pathway, the HMP shunt that you study in biochemistry. So ma'am, if let's assume if someone's G6PD pathway is working fit and fine, this means NADPH will form. So let's take your blood sample. Your blood sample has normal G6PD. This means you are forming NADPH. Today you give me 1 ml of your blood sample. I look at your blood sample under this microscope. Will your blood sample shine? Yes. Under ultraviolet light, will your blood shine? A little, yes ma'am, because I have normal G6PD and that will make normal NADPH. So my blood will shine. But now, if I have someone with G6PD deficiency anemia, jiska NADPH hi nahi bana, now when I take that person's blood and I look at it under ultraviolet light, will that person fluorescence blood shine? It will not shine. So go back to your notes now in G6PD deficiency and read that we do a fluorescence test in G6PD deficiency anemia, and now you can correlate why we do that. Because in normal people the blood shines because normal people have NADPH being formed. In deficient people the blood does not shine because NADPH is not being formed. Is that okay? How in G6PD and everything that we wanted to correlate in this slide. So most of the questions today are like that. I mean, I just wanted you to give, I just wanted to drag you to class once because I felt if I give very difficult questions, you're not giving the test. So I wanted to give you easier questions, attract you कि एक बार class में आ जाओ, so that I can discuss it and tell you what sort of questions can come in INI-CET. Because I realize that with very tough questions coming of you, many of you are thinking we will directly skip to the discussion and not give the test. So I said, okay, let's give them a confidence booster, make them give the test, and then tell them what difficult questions can come. I think this is done. This was a recent November 25 exam question. What is the contribution made by the scientists awarded this Nobel Prize? They didn't give you this image. I am giving you this image. They just told you that 2025 mein one Nobel Prize has been given. I mean, even if we try, I can also not learn their names. I don't think you can also learn their names. So they've been kind that they are not asking me their names. Just read through it once के थोड़ा थोड़ा याद रहे कि हाँ, these were the names of these scientists. तो उन्होंने हमें क्या दिया? Something about peripheral immune tolerance. What about peripheral? This I taught you in the last class only of pathology. They gave me T regulatory cell. This was one of the last questions of the last class of path 2 days ago. T regulatory cells are controlled by what? They are controlled by FOXP3 gene. So indirectly they have told me about FOXP3. If FOXP3 gene doesn't work, what was the syndrome? Excellent, IPEX syndrome. Then where did we study? Fox lady, clever lady. That was the granny. Granulosa cell tumor of the ovary, if you remember, was FOXL2 gene mutation. P53 का mutation हुआ. I mean, I will ask you two questions. P53 का mutation हुआ. Which syndrome can occur? All other questions you are telling me. Which syndrome can occur? And that is the Lee-Fraumeni syndrome. How do I identify a family is having Lee-Fraumeni syndrome? When the family is having BBBB tumors. If they say there is a family in which the mother had brain cancer, the grandmother had breast cancer, the child has osteosarcoma, matlab bone cancer, and his brother has leukemias, this means blood cancers. So when they're trying to tell me a full pedigree of BBBB cancers, I think of Lee-Fraumeni, that is P53 mutation. One more question, any virus which can go and break my P53, attack my P53, because these days the vaccination drive for HPV will get you a question in gynae or in path for HPV. HPV has two proteins, E6 and E7. E6 is the one that will go and attack my policeman. It will attack P53. E7 will go and attack RB gene. Now that they're telling you about vaccine, HPV ka kaun sa protein is responsible for vaccine preparation? L1 capsid is responsible for making that vaccine. So expect something on HPV, and tomorrow you will get questions on HPV when we do virology. तो वो तो आ जाएगा. अच्छा, ये क्या बचा? What is AIRE gene? Another INI-CET question. This is not in rapid revision. So this is always a topic which I have told students के main videos से INI-CET के लिए पढ़ लो. Anything that is to do with tolerance, central tolerance, peripheral tolerance, because now that a Nobel Prize has been given, something on tolerance will come. So this is there in, I think it will take maximum 10 minutes of your time. Main videos में immunology path में भी है, micro में भी है. Pick up any immunology from any subject, look at tolerance, it is with a timestamp, spend 10 minutes before INI-CET and wrap it up. Over there you will find AIRE gene. AIRE gene is something which is autoimmune regulator gene. It is, I'm thankful to AIRE gene. Autoimmune regulator gene is working. That is why I am not having any autoimmune disorders. The way autoimmune, the day it gets mutated, I will start having AIPE. AI to autoimmune obviously. What is PE? A polyendocrinopathy disorder. So if AI, simple na, AIRE gene is mutated, AIPE will occur, and I think this is settled. Dr. Soumya, immunology mein, I'm just telling you to watch. If you want, I'll write it here. वैसे तो 2-3 concepts, if you've seen the last pathology TND 2 days ago, it is settled because affinity maturation, somatic hypermutation, isotype switching, usme cover ho gaya. The only thing, pura mat dekho, I don't want you to waste your time. From immunology, what you should be seeing extra from the main videos is one, this concept of tolerance, and second, transplant rejection ka pathways, because in detail, if any one exam can ask you it is going, and it's tough, so it's not in rapid revision. Rapid revision में सिर्फ bullet points है, the detailed pathway of how APCs are working, how transplant rejection happens because of donor APC, recipient APC, बस ये दोनों concepts पढ़ लो, that's it. Other than that, if you've seen the pathology TND, the definitions are settled for you. In fact, this is exactly what I gave you twice. I'm not dealing with it again. In today's test also, you had a question on affinity maturation. This was only to see whether you learned it with me in path and you were up and awake and listening. I hope all of you marked it as somatic hypermutation, and this was only to test your short-term memory, recent memory. There was another question, so I'm skipping this. Anyone who's not seen path TND, we've discussed, spent 10 minutes on it, please dekh lena. Then I had another question which was from your, again, path TND only. If a patient's plasma contains elevated levels of interferon beta, what is it probably an indication of which infection? So we had studied alpha, beta, gamma, in which I had taught you that yes, mainly it is interferon alpha which carries antiviral properties. But did I tell you that sometimes interferon beta can also carry? And this is specifically a line that added कि अच्छा A तो antiviral है, but beta can also have antiviral properties. So if interferon beta is elevated, viral infection है. तो ये भी आपका knowledge test करने के लिए था. Okay. Now let's move on to the next question over here. So 2 questions that I added were purely to see whether you were able to recall something that was taught 72 hours ago, and I think you guys are smart. So there is a sputum sample that is sent, and now we'll start with all gram positive, gram stain and Zn stains. Gram positive कोका तो बोल दिया उन्होंने. Which of the following statement is false regarding the gram or the bacterial cell wall? Option is B. So firstly, when I differentiate gram positive versus gram negative, why is the cell wall of gram positive thicker? Because this is how it looks like. Overall, जो thickness है, see, if I see this also looks like say 1 millimeter, this also looks like 1 millimeter, तो thicker कहाँ है ma'am? But I am only focusing on the cell wall, and which part of the cell wall do we focus more on? Peptidoglycan. So if you see in a gram positive cell wall, the peptidoglycan over here is so thick, whereas the peptidoglycan over here is so thin. So that's the first thing that you have. Another thing that gram positive has is teichoic acid, which you can see labeled over here. What is teichoic acid? It's for ticna. Ticna means attachment. When a gram positive bacteria enters into my body, it wants to attach. So number 1 over here, teichoic acid is in gram negative. ये गलत statement था. That is why it is the answer. It is present in gram positive bacteria. अब आगे चलते हैं. What else? So number 1 positive में peptidoglycan ज़्यादा है. Ma'am, gram positive has teichoic acid. Let me get into the details of this. When I got into the details, I realized every cell wall is made up of NAM-NAG, चाहे gram positive या gram negative. NAM-NAG, NAM-NAG. It's like 2 people are sitting alternatively. The cell wall is what? NAM-NAG, NAM-NAG, NAM-NAG. This is the cell wall, दोनों bacterias का. Now, how are NAM connected to each other? These yellow dots, that is the entire gameplay over here. How are NAM connected? Are they connected or not? Over here, I can see if this is one NAM and this is one NAM, connecting them are 5 dots over here. Whereas in here, if this is one NAM and this is one NAM, there is a dash between them. No one is connecting them. When the 5 dots are connecting them, they are called as pentapeptide bridge. 1, 2, everywhere you will see 1, 2, 3, 4, 5. That is seen in gram positive. That is not seen in gram negative. तो ये भी पूरा हो गया. Hence we always study ना PPPT. Positive में peptidoglycan, positive में pentapeptide bridge. Positive has teichoic acid. Then what does negative have? Negative में कुछ तो होगा. Negative predominantly has lipopolysaccharide. That gets you a lot of questions. Listen carefully. When I say lipopolysaccharide, it is supposed to have something lipo. It is supposed to have polysaccharide. तो ये तो याद रहेगा. Learn the third thing. The third thing is the O antigen. Before I start, where is O antigen studied? In a test for typhoid. When you do the test of typhoid called Vidal test, you study 2 antigens. One is the O antigen, one is H antigen. O antigen abhi kar liya, cell wall, because typhoid is a gram negative organism. And H antigen is for flagella, which I will be teaching you in 5 minutes when we come to questions. So now coming to O antigen, in fact, the most variable part of any gram negative cell wall, because you will say that, ma'am, if every gram negative organism has the same antigen, then all the bacteria should be the same. Why are different gram negative bacteria there? जब same antigen है, तो then why did you waste 3 hours of our life teaching us gram negative organisms? वो तो same थे, वो नहीं same है. Antigen name is the same, but the antigen has many variations, and every gram negative organism has a different O antigen. E. coli maybe will have a different antigen. Then when I come to Salmonella, that will have a different O antigen. Shigella will have a different O antigen. So this is a very variable part, and hence all bacteria are different from each other. Coming to the next, of course, it is going to be the next one, that is polysaccharide. And lipo kahan gaya? That is part A, that is lipid A. And do you know what is the favorite property of lipid A? It has endotoxic activity. That is why we study that gram-negative organisms make endotoxins. Where is that endotoxin is responsible for a lot of sepsis and a lot of big severe clinical features, na, septicemia, etc. So where is that endotoxin hiding? It is in lipid A. How are they arranged? They are arranged as OPA. So first O antigen hota hai, then polysaccharide, then it is going to be lipid A. So I guess this is also settled. Let me go back and see if I can answer the question. Is region 2 of lipopolysaccharide the polysaccharide? Region 2 matlab OPA. Yes, region 2 is the polysaccharide. So this is also correct. Ye bhi correct. In lipopolysaccharide, the O antigen specificity is what is O antigen's work? Yes. Every bacteria has its own O antigen, so specificity will be based on O antigen. This is also correct. I think I've solved the question, and in that process, I've done most of the questions of this. But one more question, I'm just going to enlarge this picture a little more for you to understand. This is an image that I've added in the next version of rapid revision, and as I said, I'll be teaching you all those additions here only. So then I realized that a question zyada aa raha hai, which you must have seen, called periplasmic space in your GTs, right? So now just understand, why did it get formed? Gram positive mein to there's a very thick peptidoglycan. Gram negative में that has been squeezed into a very tiny peptidoglycan. तो मैम बाकी का space किसने लिया? There must have been some space that is created if you look carefully. This is gram negative cell wall. I've just got you a very big picture. This is probably the peptidoglycan that we are trying to show you over here. And this is a combination. This is actually a picture that I've got you for an organism which is having properties of both, to make you understand कि कैसे बना periplasmic space. Over here the peptidoglycan is so thick, I thought it is gram positive because very thick peptidoglycan. Then I could see teichoic acid, then also I thought, oh, it is gram positive. But then if it is a feature, or you've heard of this last chapter of microbiology, kya hai? Gram poor organisms. What are the gram poor organisms? If you tell me, then I'll be able to explain this to you. The gram poor organisms were MRCS, Mycoplasma, Rickettsia, Chlamydia and spirochetes. Ma'am, they are gram poor organisms. Means they have properties thoda of both. Little like gram positive, little like gram negative. So then till here I thought this was peptidoglycan teichoic, I thought it was gram positive. But then I saw there is something called periplasmic space. Now that is not a part of gram positive, that is a part of gram negative. Periplasmic space gram negative में होता है. तो क्या है periplasmic space? Inside it I can see something like this. कुछ आपने ऐसा बनाया. What is inside it? Flagella. But flagella is supposed to come out and make the cell move. I am saying the flagella is inside. What is periplasmic space famous for having? It is famous for having a flagella inside called endoflagella. And endoflagella is seen in which bacteria? Spirochetes. So you might want to start off by saying, "Arey, this is peptidoglycan and teichoic acid, ये तो gram positive है." But when you saw periplasmic, you said, no, this is mixture. This is gram poor organism with this endoflagella. This is spirochetes. Yes, that same Treponema pallidum, syphilis wala truck driver wala story, that could come with this kind of a picture. So you've understood why do I call it gram poor? Because it has properties of both, and periplasmic space has endoflagella, which is another important question. Can we move on to the next question over here? Something again not in rapid revision, hence I added. Arrange the layers of spore. I'm sure spores you have read many a times in your rapid revision. But now from main videos, I wanted to tell you the layers of a spore. How does it look like? Spore is what? It is the sleeping form of a bacteria, the dormant, and it is very dangerous. In entire sterilization, I'm trying to kill the spores all the time. कि bacteria तो हो जाएगा kill with even alcohol or acetone, but spore is something that I want to kill. What are the layers of the spore? If I would have just, let's imagine this question came in the exam, I was a student considering INI-CET, I wanted to attempt maximum possible. अगर मुझे करना होता तो if they said from the correct order, core तो अंदर ही होगा, right? Core will always be inside. This is basic. एक option rule out हुआ. Then उसके बाद something that sounds like an exit will always be outside. तो चलो फिर अब बचा एक और option rule out हुआ. Exit वाला बाहर होगा something. core will be inside. Now whether it is cortex outside or coat outside, so I will always wear a coat, है ना, like I'm wearing a blazer, a coat. Same way you will always wear a coat outside and then everything else will be inside. Same way this is the answer, and that is exactly how I have learned the layers of a spore. A spore in the center will have something called a core. A spore on the outside will have something that sounds like exit, so exosporium. Then I'm left with coat and cortex, so obviously the coat will be worn outside and the cortex is going to be inside. So 4 layers yaad ho gayi. I hope that is settled. Please note the core that you have, this core is going to be, it's hiding inside na, to ye heat se bach jata hai. It is heat, it is what makes the spore heat resistant. Why do I say that don't have water only by boiling it? Why do we have so many water filters in our house when I could just boil water for 15 minutes and drink it? Because does boiling of water at 100 degrees kill the spores of a bacteria? It doesn't. Why is the spore heat resistant? Because of which part? Because of the core. What is present in the core? Dipicolinic acid, which makes it heat resistant. These are extra pointers not in your rapid revision. Hence for INI-CET, maybe you can give them a skip for NEET later on and I wouldn't mind it, but for INI-CET please ye pad jao. And that is why, you know, if they just want to correlate that, oh, there is a village in which you have to, you know, tell people about how boiling is not enough and they should be doing filtration of water, etc. So this is exactly how they would correlate that. Why is the spore not getting killed? What makes it heat resistant? Dipicolinic acid. Which part of spore is it present? It is present in the core. Now let's talk about further. This coat is also the reason it is chemical resistant, and that is why I say now that so many chemicals cannot kill the spores easily. Even if I'm using chemicals, I have to use them at a higher concentration so that I can kill the spores. So chemical resistance is based on coat and heat resistance is based on core. I think we can move on to the next question, where we have the correct statement regarding Fimbriae or pili. I give this in every TND because this is a very INI-CET type of a question. Okay, first they'll give you 5 confusing options, then you'll have to select out of it. So now tell me, you might want to feel Fimbriae are made up of fibrillins, but no, Fimbriae are actually made up of antigens and Fimbriae are made up of, Fimbriae का क्या दूसरा नाम है? पाइला है ना, so they are actually made up of pilins. Fibrillins से नहीं बने होते, ठीक है. Pili are involved in bacterial conjugation. Yes, I had studied. One bacteria wants to connect with another bacteria. It is going to connect as what? By that Fimbriae or pili. तो ये तो मुझे correct statement ढूंढना है ना, ठीक है. So this is how I mark in the exam, that I keep in my mind the second statement is definitely correct, and you will also notice it is there in every option over here. But like I said, you don't have to jump to the option, अपना answer बना लो. Pili can be involved in motility through a process called twitching. Is pili for motility? Because I had studied pili and Fimbriae for attachment, but there is a special type of a pili which is called as type 4. This is also an INI-CET PYQ. एक special pili है, type 4 pili. Other pilis are involved in attaching the bacteria, but type 4 pili is responsible for motility. And when I say motility, कौन से दो bacteria के लिए did they ask you? Type 4 pili is present in Neisseria. I don't expect Neisseria to be motile. Neisseria तो is a cocci, ma'am, gram-negative cocci. Cocci कब से motile होने लग गए? Hence INI-CET asked you that is there a special type of structure that Neisseria has? And the second one, even pseudomonas. These two questions for INI-CET have been asked, that इनका motility किस पर based है? Their motility is not based on the flagella. It is based on the type 4 pili. So yes, pili can be involved in motility. I think as of now ये भी ठीक है. Okay, now coming to the next, the Fimbriae play a role in pellicle formation. Again, this entire question was only given based on INI-CET, and if after this test you felt कि मुझे इतना information नहीं आता, many of you will start having that doubt that should I now skip to and jump to my main videos and start reading 45 hours of microbiology from main videos. Right now it will be a very tempting thing to do. You say I'll take out 4 days and I'll wrap it up. But the problem with microbio is when you read the full videos, na, you can't retain them for a very long time, and that is when the core that you had to learn, that is also lost, and that extra useless information which you were learning, that is also not going to get retained. So that is, hence I say that आपका जो पहले 14.5 or 15 hours of rapid revision है, that becomes your baseline, and baseline as in 85%, 90% उसमें sorted है. Now that extra 10% के लिए, do you want to spend looking at 30 hours of video lectures, or that extra 10% you want to do from TNDs and you want to do from the Q bank, which seems to to be a more smarter approach, and that is why I'd never get tempted. Micro का नया question तो दिखेगा, tough अजीब अजीब रट्टा questions, factual questions will come up, but the rule is that you stick to that 90% of your core and then just add that 10% useless knowledge if the examiner wants to confuse you. So let's go on to the next. Dr. Soumya, INI core topics are the ones that I have covered in this, but I will still be giving you a list, but not right now. Abhi you go through these 2 sessions, and after this, tomorrow, I will let you know all the topics जो extra अगर कुछ पढ़ना है, right? Okay, coming to the next. Yes, for Fimbriae, we do a test called pellicle formation test. नाम याद करना है, surface pellicle test. That's the name of it, and that's all that I have to learn. Surface pellicle test. ठीक है? That's just what is the test for Fimbria. Coming to pili, are longer and more numerous than flagella? No. If you remember the bacteria ka structure, the longer one is always the flagella and the pili are always chota wala. So they are not longer. This is wrong. The only 3 correct statements that I got were 2, 3 and 4. Were 2, 3 and 4. Is that okay? Yes. Pili ka sab kuch samajh aa gaya. Especially if I expect a question is on type 4 pili. So take home message. All pili are for attachment of bacteria, but one pili for motility is a type 4 pili present in which 2 bacteria? Neisseria and Pseudomonas. आगे चलते हैं. Now a lot of these match the following 2-3 questions were like this. Let's start with Neisseria. ये तो अब हो गया ना? I just told you Neisseria has what for motility? Organ of locomotion. It is type 4 pili. Okay, Belantidium, coli and parasitology we always used to study as celi. So this is also settled for us. How can you forget Trichomonas vaginalis? And can you tell me T for Trichomonas vaginalis, T for which type of motility? Twitching motility. And Trichomonas में कौन कौन से forms होते हैं? Trophozoite होता है, cyst होता है या both? It only has trophozoite form. So Trichomonas, the strawberry cervix वाला, that is going to be twitching motility. That is going to have trophozoite only, and it is because of a flagella. Toxoplasma gondii का तो I don't read any motility, only it doesn't have any organ. So I think by now you were able to solve this easily. Let's go to another match the following more motility questions. Now just now I taught you twitching motility. किससे mark करोगे? Trichomonas से mark करोगे. So that is done. In the last class also I was teaching you गिरता हुआ leaf, falling leaf motilities for which one? Giardia lamblia. How can you forget INI and FMGE? दोनों का favorite question. Tumbling like a drunk friend falling left and right. Who is that drunk friend bacteria of yours which is falling like a drunk friend? Listeria. But now there's one more thing. Will it fall and tumble at every temperature? Or does that change as per the temperature, and that is what you call as differential motility? Can you tell me the temperatures? Will it tumble? I'll write T for tumbling motility. It will tumble at 22 to 25 degrees and it will not tumble at 37 degrees. And how did we learn this? Tumbling sounds like getting drunk. You can afford to, I mean, not promoting this and don't follow it, but you can afford to get drunk at the age of 20s because your liver is young and healthy and ready to support you. But you can't afford to get drunk by this age because by now your metabolism and your liver has pretty much slowed down. So you can tumble in your 20s, but you can't tumble in your 30s, and that is what life lesson Listeria teaches us. Coming to the last one, darting. Darting is the one that going very fast like a star, and star wala is shown by Vibrio. Any other organism other than Vibrio that can show you this? It is known as excellent Campylobacter. Campylobacter also shows you the darting motility. Yes, Dr. Akash, I totally agree. General microbiology is little tough. Little nahi, you are just underplaying it. I think on my face you are saying it, but I would say it very openly. It is very tough. Not just micro, general, overall micro is a very tough topic. Not because ऐसा कोई बहुत high rocket science concept आ रहा है इसके अंदर. It is only because it has facts, and it has facts left, right, center. I have seen so many people trying to give this a conceptual angle, which comes. In medicine, maybe the conceptual angle comes, but it never comes in the micro segment. वो जो conceptual angle आता है, वो आता है कि अच्छा, it is a patient and infective endocarditis is happening and all this blood culture is positive, but the last line is ultimately a fact and a ratta that अच्छा, which is the bacteria causing it, and you cannot put any sense into it. It does not, either it comes with clinical practice, which you will do later on, and then you won't need all these mnemonics. But right now, when you're not 10 years into your practice, right now it is a lot of ratta. So those too much to ratify. It is even I at times feel it gets worse than pharma also. And I am totally with you guys on this. Hence we make those silly mnemonics, those silly stories. And if you are not a mnemonic person, that makes it even worse for you, because there is no other way to learn it. I mean, there is nothing that I will put a concept into all of these motilities and I will find some new logic behind it. वो है ही नहीं logic. तो आपको रटना पड़ेगा. So if you are good with ratta, congrats. But if you are bad with it, then revision is what is going to save you. When you do video lectures, sorry taking 2 minutes extra of your T&D, but when you're doing video lectures, you might feel that I have completed my syllabus, but it's actually the tip of the iceberg that you've touched. Because by looking at video lectures, you're getting yourself pseudo confidence कि मैंने सुन लिया, and सुनने से तो everyone, like, don't you get that dopamine kick when you're watching too many Insta reels or YouTube shorts and listening to those podcasts and you feel you're very knowledgeable? But if I make you sit in front of someone and say that, okay, now discuss, that is when the real you comes up, right? Your real intellect shows up. So same way, when you're doing video lectures and listening to these stories and doing haha, giardia, lamblia, trichomonas stories, you're feeling very happy about it, but you've just touched the tip of the iceberg, because now when you'll open the Q bank, that will be like opening a can of worms. That is when it will become very messy and you will get to know that अच्छा ये तो मुझे आता ही नहीं है, because I have studied it, I have laughed over it, but when it comes to recalling it with a question is becoming difficult. So the rule for micro is 3 times over. After shutting the video lecture, you have to revise your notes. I'm not saying the same day 3 times, spaced 3 times. Pick up 3 such subjects, which I say as micro, biochem and pharma. If you think video lectures and 1 revision will get you through, answer is a big no. You will have to do 3 revisions for micro, biochem and pharma. And those 3 revisions have to be at least 7 to 10 days apart. Spaced revision. Your mind forgets, you read again. You forget, then you read again. So when your third cycle of rereading comes, is when you'll get some kind of confidence कि हाँ अब हो जाएगा मेरे से. That is how it works for these, okay? हाँ, there are anatomy to PSM. To PSM तो I mean, I shouldn't say this, but वो एक give up वाला phase आ जाता है ना, that even if I revise it 3 times over, the examiner will give me some yojana or some biostatistics question where I will get stuck. Biochem, micro and pharma के साथ confidence ये है that 3 बार कर लिया तो at least 90% आएगा. तो PSM के साथ तो वो तब भी नहीं आता. So PSM you take with a pinch of salt, that I will put my entire life into studying PSM, all my brains, my intellect and my energy into it, but then I will get 2-3 questions in the exam which I will not be able to solve. So if I go with that mindset, I will not trouble myself during the exam paper. I won't get demotivated. तो वो ऐसे ही सोच के जाना. Yes, bacteriology stuff वो सब है, but as I've told you, applicable to all parts of micro, you will have to revise it 3 times over. You do it from any source, my videos, any other videos, and I know for micro, biochem, you try to find the shortest. So many people have told me that, ma'am, 15 hours rapid revision is too much. Can you please make it into 8 hours, 6 hours? But for that, you have LRRs and all, which are so easily available on every place, whether YouTube, Telegram or whatever. But then for that, but does that LRR ka 5 hours make you solve all 90% questions? Answer is a no. How can 5 hours make you solve 100%, 90% questions? वो किसी भी subject के लिए नहीं हो सकता ना, for a big subject. So then that is why I say कि 15 hours तो पढ़ो, don't read 45 hours. But 15 hours को और छोटा मत करो. And if you want to make it concise, make it so before the exam 10 days. 10 days before the exam, you switch to only this session or only LRR, I'm okay with it. But right now when you have a considerable weeks in hand, this RR तो minimum, उसको और छोटा नहीं करना, because then you will not be able to sail through in the paper. So I might sound a little toxic in telling you कि नहीं 15 hours पढ़ो, but then I could have been more toxic in telling you 45 hours पढ़ो. I have come some way, you will also have to walk some way. We have to meet in middle ground na? If I have come from 45 to 15, you will also have to come from 5 to 15, and if we meet at that 15 wala comfortable number, I will be happy after the exam that I contributed, you will be happy that you got your rank. That is how it works. It's a mutual walkthrough, right? Coming back, एक motility इसमें नहीं था, that is everyone's favorite, swarming, which they want to ask you in INI-CET. I think I've given you enough motivation to survive through the next 2 hours, right? That I've done. But then motivation has a half-life. The half-life is after the lecture, your motivation will go away. So I hope it remains, and that I've told you how to do it. Spaced revision करना है, एक हफ्ता 10 days छोड़ के. Coming back, swarming means this round thing happening, and that is going to be by PVCs. अब INI-CET में P for Proteus तो पूछ ही लेंगे, it is V से क्या क्या आता है, and Vibrio is not Vibrio cholera. Vibrio cholera तो अभी you told me ना, the darting motility comes. Vibrio कौन सा? Vibrio parahemolyticus. What kind of a history do I get for Vibrio parahemolyticus? You said ना bacterio tough है, तो कल भी करेंगे bacterio, थोड़ा आज भी करेंगे bacterio. Vibrio parahemolyticus, I always say P for parahemolyticus, P for prawns. Basically something to do with the food poisoning that is after consumption of seafood. P for parahemolyticus, P for prawns. ऐसे याद रह जाएगा. Vibrio एक और है alginolyticus. This sounds like Ajinomoto. Ajinomoto जो Chinese food में डलता है ना, that makes it very salty and spicy and tasty also, which we should never have if we have hypertension and all these problems. So that Ajinomoto salt always reminds me, this is the most tolerant to salt, even more than 10% of salt. When every bacteria will die, this Vibrio alginolyticus will say, I am to surviving because I love Chinese food, I love that salt. So it survives that. It हाँ आँख कान से धुआं, because Chinese food खा के that is what happens ना, there is धुआं coming out because it's very spicy. So आँख कान से धुआं means these patients will have otitis media and these patients are going to have conjunctivitis. That is what alginolyticus causes. Survives at a high salt, आँख और कान से धुआं. So आता तो है. Why do you think that it's tough? All of you know it. Stories will come back. हाँ immediately नहीं आएगा, but exam में जब question आएगा तो वो कुछ something will come back in your mind. Trust me on that. I have seen so many batches. सबके साथ हुआ है तो you are no different. It will happen to you also. Coming to the next, PVCs, Clostridium tetani and Bacillus cereus. Cereus का तो we know, the real Chinese वाला food poisoning. Chinese fried rice वाला food poisoning, how can we forget? Where the incubation period is less than 6 hours. Someone had fried rice from a stall or a street side food vendor and uske baad within 6 hours food poisoning, Bacillus cereus. Finally coming to Serratia. Why is Serratia important? Serratia always makes a red color pigment. Someone coming to you with pneumonia and a lot of cough, a lot of sputum. The sputum has a red color pigment to it. Red color pigment, always think of Serratia. So all of these PVCs, they go round like this. Now you say, I want to stop this. I don't want all this round to go. Inhibit karo swarming ko. So I'll say, okay, usually the agar that you use, उसमें कितना % agar होता है? Whether it is blood agar, nutrient agar, ABCD, कोई भी agar, I use a 2% agar. But if you want to stop this round movement, I will now use a firm agar. Firm agar uses 5 to 6% of agar. What is the concentration? I have basically firm के लिए 5 to 6%. I have made this very solid. If I make it very firm and solid, how will this bacteria roam around freely? It will not, and swarming will be inhibited. 5 to 6% agar use करा तो that is firm agar. अगर usual करा तो usual 2% agar. So I think we are settled with all of these questions. I move on to the next. ये तो simple one liner था. Which of the following does not contain a polysaccharide capsule? All the capsules are made up of polysaccharide except Bacillus anthracis, which is made up of polypeptide. Now which peptide? That is also another way of asking you. NEET PG तक तो polypeptide capsule लिखते हैं, but in INI-CET they like to ask you which polypeptide? Polyglutamate capsule. If this is also written, वही peptide ही है ना, it is something to do with amino acids and peptides, so it's the same thing. That is what it has. You know, Bacillus anthracis is different in every way. All capsulated bacteria, they have polysaccharides, but anthrax says no, I will to have peptide. Then do you know all the bacteria, their toxins, they are going to have 2 parts. All the toxins are bipartite, but who will tell me what are the toxin of anthrax? Anthrax says my toxin is going to have 3 parts. That I have told you. You will tell me the 3 parts. Very good. थोड़ा सी bacteria कल के लिए, but today we are doing bacterias. EPL, English Premier League तो हमारा favorite mnemonic है. जब कुछ नहीं समझ आता, we add this. I'll go back to pathology and ask you. There were 3 selectins that we studied. Remember inflammation chapter, selectins. What are the 3 selectins? E-selectin, P-selectin, L-selectin. What are the 3 parts of the toxin? Quickly asking you. One is known as, ma'am, edema toxin. The other is protective toxin. And the third is what they write in the options. Edema toxin सबको याद रहता है. Protective toxin you remember. L से they write in the exam lethal toxin or labile toxin. And think very as a sensible human being कि anthrax is a category A bioterrorism agent. Is it going to be very labile or is it going to be lethal as hell? So of course it is going to be lethal. It is known as the lethal toxin. So जब अगर आप mnemonic में फंस जाओ, which happens it happens very commonly in micro, that EPL याद हो गया, but what with L? That's how our mind works. But then put your logic into it. I think we are settled. Moving on to the next. Don't go by these numbers. I've changed the order. I'll discuss all 50 by the end of it. Here we have a 45-year-old male with fever and eschar on his arm and handling animal hides. As soon as they say this, I am of course thinking of hide porter disease. Someone who is handling animal skin like a coolie, carrying the animal skin, and we are of course using Bacillus anthracis. Now what is the special stain that I will use? Excellent. McFadyean reaction. So now if you look at carefully, all the 4 options mentioned over here are for handling anything that has a capsule. Can you tell me for which fungus? To know the capsule of which fungus do I use India ink staining? Today we have a question on this coming up shortly. That is Cryptococcus भी capsulated है मैम, उसके लिए we do India ink. Okay, latex agglutination test I can do for any capsule because agglutination means I'm trying to look at the antigen. So for any capsule antigen, especially for Cryptococcus only, we do the latex agglutination test. Now coming back, ये दोनों reactions. If they tell me anything to do with all of the polysaccharide capsules, I told you all capsules are made up of polysaccharide, I prefer doing a Quelang reaction. But for anthrax, which carries which kind of a capsule? Polypeptide capsule, as you see over here, I prefer to do McFadyean reaction. So now my question is, what is McFadyean reaction? यहाँ से level 2 question क्या आएगा? Okay, this is a case of anthrax, animal history है, animal skin के साथ anthrax है. You are doing McFadyean reaction. How do you do McFadyean? This is what INI-CET is ना, कि lab में जाके पूछो how is it done. So I will be taking the bacteria and I am right now assuming कि bacteria has a capsule because it is anthrax. I will be adding what? Polychrome methylene blue. Excellent. We have studied M for M. McFadyean reaction में क्या use होता है? Polychrome methylene blue. Where else have we studied methylene blue in pathology? कहीं तो पढ़ा था न्यू मेथिलीन ब्लू, हिमैट में पढ़ा था 2 days ago, one of the last questions. न्यू मेथिलीन ब्लू is a supravital stain for reticulocyte. दो ही तो है ना, methylene blue and then the story finishes. New methylene blue was a supravital stain for reticulocyte. Polychrome methylene blue I am using in the McFadyean reaction which I am doing for Bacillus anthracis. What about the Quelang reaction? वो story याद है? Quelang में swelling, quelling reaction में swelling होगा. So now when I say Quelang reaction, it is going to have a swelling of the capsule. Why swelling of the capsule? Because this is an antigen antibody reaction that I am going to do. Very good. Dr. Akash says, ma'am, methylene blue एक और जगह आता है, acid fast में बिल्कुल आता है, बस that is exactly why I didn't mention, because I was waiting for you to say that in the next question. Here we come to this, where a 32-year-old laborer working at a construction site comes with fever and hemoptysis. In our country, if someone is having fever and blood in the sputum, either I think of TB or I think of cancer. My first 2 common things come. Over here sputum sample लिया, दिमाग में TB cancer चल रहा है. If they shown me this picture, of course it is to do with TB. Micro का TND है, of course it is to do with TB. So I am doing the ZN stain. For ZN stain, what were you saying? कि मैम methylene blue वहाँ भी आता है. Exactly. That is what I've got this. That's why I put this as the next question. तो क्या है ZN stain में? Firstly, look at this picture. If I just tell you to, is this positive for TB? Yes, ma'am. You can see so many of these red color TB bacterias. So many of them are noticed. I can see, chalo, let's say I can see many more, but I will not torture you. Let's say you can see 5 of these. ठीक है? Is this TB? Of course. If you would have only seen 1, is this still TB? Yes, the rule is that even if 1 bacteria is seen, TB program में भी ये लिखा है, even if 1 bacteria is seen by the microbiologist or pathologist, it is a case of TB. So never wait for 10, 20, 30 to be seen. Even 1 is going to be TB. But now why does this look red in color? And why does the background look blue in color? The red color that comes is due to carbonyl fusion and the blue color that comes is due to methylene blue. मान लो अगर एग्जाम में नहीं भी समझ आ रहा था तो ये ग्रीन, वायलेट, टैनिक एसिड ये सब तो वैसे ही रूल आउट कर देना चाहिए था ना. You didn't have to learn the mnemonic for this. You just had to put your common sense. And in one INI-CET exam, this is a little purana PYQ 4 years ago. They didn't give this picture or this. They just asked you on ZN stain, organism is dash in color and background is dash in color because they they just wanted to test, have you ever seen the picture without giving this image in the exam? Organism is red in color, background is blue in color, and that is how we learnt it. So what is this class, has asked mnemonic. So you know कि सबसे पहले red color डाला था. The primary stain is going to be carbol fuchsin. What is carbol fuchsin? They don't write carbol. Carbol is a type of a phenol. चलो मान लो they didn't go as per your mnemonic ना, but they wrote phenol. तो carbol is a phenol, तो वो भी चलेगा. That is also right. That is giving the red color. After that I do heating. What is heating? Heating is going to act as a mordant which will fix this red color even more. After that I will remove the red color, and that is going to be by acid alcohol. Which acid do we use? Sulfuric acid use करते हैं मैम, H2SO4. and TB था तो कितना परसेंट यूज किया होगा? TB के लिए 20 to 25% यूज करते हैं. T20 TB, T25 20%. and finally the background has to be blue, so I have added the secondary stain which you were saying is methylene blue. So carbol fuchsin, heat, acid alcohol and methylene blue. Then after this you don't even need the mnemonic ना, वो तो दिख ही रहा है. कुछ red color है, कुछ blue color है, then heating and then decolorizer. But now if I ask you what percentage of H2SO4, so everyone fill in the blanks from, consider this as a workbook of your rapid revision. TB में कितना percent? Very good, 20-25. Lepra meanwhile everyone had answered कि अच्छा lepra 5 alphabets, तो 5% H2SO4. Now what will this be known as? This will be known as the fight for acostain. 5 fight for acostain, 5% H2SO4. Same है, it is the same Zn stain, बस नाम change कर दिया because I used 5% H2SO4. Controversial questions. Coccidian parasites may be 5%. There are some books, there are some apps which have written 0.5%. कुछ जगहों पे है printing error. So I'm telling you upfront live that Coccidian parasites के लिए please 5% मानना. Now coming to Nocardia and Legionella, 1%. This is where I expect an INI question. Nocardia causes what? We will study tomorrow in bacteriology के pulmonary infection कराता है. And उसके लिए what percentage do we use? 1% H2SO4. Now I want to call it a modified Zn stain. Just the name modified added. So you know they give you a question that there is a person having pulmonary infection suspected to have Nocardia. सब लिख दिया. You want to now confirm Nocardia with a special stain. What stain will you use? यहाँ तक everything was told. Option A was Zn stain, which most students marked. Option B was modified Zn stain, which you should have marked because that is how they play. Whenever answer is option A, I'm not saying it can never be the answer. I'm just saying that just read it twice over के B, C, D options पढ़ लो एक बार, because many a times they just want you to be a little hyper in marking it and they give something very obvious in option A which every student would want to click. So remember Nocardia के लिए the better answer. Why modified? Because I've used 1% H2SO4. Now coming to spores and sperms, it is 0.25 to 0.5% H2SO4. This is settled, which brings me to the next question in today's test. Which of the following organism listed below is capable of being stained with acid-fast staining? So was Nocardia part of it? Yes. Leprosy, बिल्कुल yes. Actinomyces तो नहीं पढ़ा. Okay, what is this? Cryptosporidium and Isospora. These are the coccidian parasites. So 1, 2, see first I made my answer. मेरे हिसाब से 1, 2, 4, 5. These are all acid fast. Do I see them in the options? Yes, I see 1, 2, 4, 5 in the options and hence I mark it. So except for actinomyces which is not, अब सबका वो भी बता दो. Tell me the percentage. In Nocardia 1% अभी you told us. In lepra ma'am you told us fight for acostain. And for the coccidian parasites you told us it is going to be again 5%. But now I want to add something. In coccidian parasite, the only change is not 5%. The change is that I don't heat. I do carbol fuchsin, then I do decolorizer, and then I do methylene blue. I skip this test. So in coccidian parasites, can I say I do a special type of stain called the cold Zn stain, also known as Quignon stain. Tell me the third name for INI-CET other than Quignon stain. It is known to be the Gabbett stain. Cold Zn stain, Quignon stain, Gabbett stain. Same cheez कर रहे हो ma'am, but cold है ना. You are not doing the heating, but heat was a mordant. You mean to say I'm not fixing the stain? I have added carbol fuchsin who will fix that red color because you are not heating. What should I do? How should I replace? Then in that situation, because you are not heating, you will increase the concentration of phenol, and that is what they give you as a question. That if you are not using a mordant, then what do? I increase the concentration of phenol, which is carbol fuchsin only. So I think we are settled with all of these. I can move on to the next staining. Microbiology resident wants to do gram staining, prepares pure cultures of Escherichia coli, which is gram negative, and Staphylococcus aureus gram positive. On adding the primary stain and mordant only, which of the following observations would correctly define this? What did you mark over here? I hope you didn't say that staph is gram positive, so gram positive is going to be purple, and E. coli is gram negative, so gram negative is red or pink, and that is why you marked B, but that end result to yahi hota. But here I said you have not done the end, you have only your midway, halfway through gram staining. In halfway through gram staining, both the organisms will come out to be purple. So listen carefully. You are doing the gram stain. I think this mnemonic is so universal in every place. This is how you read it. Come in and stain. What is the first thing that you add? You add crystal violet. When you add crystal violet, the gram positive also becomes violet. The gram negative also becomes violet. Abhi both are violet. Both are crystal violet. After that you will add iodine. Iodine is what? Iodine is the mordant. Fix kara to yahan par bhi both of them are going to still be purple, and that is what I told you ki yahan tak kara hai. You have added the primary stain and mordant only. Now both the organisms at the end of this stage are going to be purple only. But then comes the most crucial step, the decolorization step, where you are adding that alcohol acetone and removing the color. Yahan par hi to hota hai difference. That is why this is the most crucial. Here is where you decide, oh, such में it is gram positive or gram negative, because till now everything was purple only. On adding the decolorizer, the gram positive ones will say we will to still keep the purple color, and now you know why. Because gram positive had a very thick what? It had a very thick peptidoglycan. So peptidoglycan catches that purple and then catches it for a lifetime. From the start till the very end, gram positive hamesha purple rahega from the start to the end. Once you put a purple color, peptidoglycan says, I am not letting it go. Gram positive ki to life set hai. This gram negative initially was purple, but on adding a decolorizer, gram negative says, look at this, it has become white, it has become colorless. उसने अपना कलर छोड़ दिया. Why? Because gram negative had lipopolysaccharides na, OPA, तो वो lipid के through the color nicely goes out, permeable, the color goes out. This purple color has walked out. Now I will have to find another color, and that is why the second color that I add for gram negative organism is safranin red, which will give it that reddish or pinkish color. So now you've understood the cell wall, you've understood the gram staining, and right now I said that okay, abhi sirf pehle do steps kare, so both of them are going to be purple. Is that okay? Yes, everyone? Can there be a situation where if I add primary stain, if I add the mordant also, and I add a decolorizer and stop it over here, and I say that because I was very new and it was my first day of residency, I ended up adding excessive amount of alcohol or acetone, more than required, excessive amount, or I forgot the stain over here, I forgot for 30 minutes, 40 minutes, and I went for my lunch break. Now when I came back, what will be the result? सबसे crucial step था, means you had to time it properly. The concentration, the timing, everything had to be kept very done with a lot of dedication. If you were a little, casual with this stage, everything would have become colorless. See, gram positive is saying I will keep purple, but excessive amount of alcohol dal doge for 30, 40, 50 minutes you will put alcohol, then even that gram positive will give up. It will end up getting destroyed. Its cell wall will end up getting destroyed. And that is why both of them will end up becoming colorless. And hence I'm trying to tell you, sabse crucial step, what you have to time, what you have to be extra cautious, stand on the head of the slide, not read it, not leave it and go anywhere for lunch or coffee, that is the decolorization.